And let me see if
Good.
I can. Oh,
July. Right. Just make sure recording in progress. The July focus group for the Knowledge Portal Network. I love these. These are actually a chance for us to get very focused on the different types of tools and features that we're previewing into the Knowledge Portal over time that are in sort of like the beta state and being tested and developed as we speak, with data within the confines of the Knowledge Portal Network and the broader consortium. They all work together. Really happy today that Laura Scott and our to walk us through a new browser called Colocus, from the great group that brought you Locus Zoom, which is a gold standard in our field, and many other visualizations. This is a new visualization that we're hoping to be, a molecular trait QTL and colocalization browser to support the identification of potential causal genes for cardiometabolic diseases first, and then broadening to all of complex disease. So today she's going to preview this for you, talk about what we've done so far in this space, some of the challenges, some of the goals going over time, and also ideally really get your feedback on where we are and what would be valuable as we develop this for prime time on the Knowledge Portal Network. So as per usual, we'll be recording this, and if you have questions, you can either pop them directly in the chat, and we'll be monitoring them so that we can work, directly. We want this to be interactive, not just a presentation, but someone talking to each other and giving us active feedback. You can also put them into the chat or or raise your hand, and we'll adjust them as we go. And with that, any other programming notes before we begin? All right, super. I will hand it over to Laura. Thank you, Laura.
Thanks very much, Noelle. Yeah. All right, can you see my screen here?
Looks good.
Great. Okay, so what I'm going to tell you today is about a browser that we've been developing that, as Noelle said, kind of grew out of the visualization of Locus Zoom, which allows you to see, the relationship of the strength of a p-value for a SNP, to a molecular trait over a specific part of the genome. So this tool has been developed in strong association with Ryan Welsh, originally a lot of the original things from Andy Boughton, and now Jeff Holtzman has joined us, and together with Mike Benke, there has been an incredible amount of beta testing that I want to acknowledge from Sarah Brotman and Elaine Broadway out of Karen Mulkey's lab in the initial data that I'll show you some examples of today. And then, of course, with feedback from the Broad Portal team also. So I've written up here a trait, QTL, CMD, GWAS, but this could be applied to any, and hopefully will be applied to many different types of GWASs to aid in identification of potentially causal genes. Okay. No, sorry. Okay, so I'm going to talk to you today about the rationale of the colocus development, show you a look at the browser as it currently is, talk about how we're integrating work between the different parts of the AMP CMD that we're part of, FNIH and the Knowledge Portal, as that may be useful for thinking about in other groups that might want to undertake using this, a little bit of coming attractions, and then a summary at the end. Okay, so to bring everybody up to speed, when we're talking about colocalization between a molecular trait and a GWAS, what we're talking about is, does the pattern of SNP associations in a given region, is it consistent between a GWAS and a SNP and a gene? So here, if you start on the bottom, you can see a picture of association of SNPs with expression levels in the CBX3 gene. So across here, you're going to see lots of these plots, is the chromosome position. And then each one of these points represents the testing of a SNP. And this is the negative log 10 p-value. The higher up you are, the stronger your association. So this is a beautiful example of a strong association. There are no other variants around. You can see this is the LD scale, kind of the redder it is, the stronger LD. As you go to purple, the weaker LD. This is a beautiful variant all by itself, potentially causal. If you look at the BMI meta-analysis again, you see this is a perfect example. I'm starting with, of course, a very strong GWAS signal greater than 10 to the minus, right, 40. Again, a single variant. These signals look very much like each other. These, you would say by eye, and if you had to, statistically, look like they're colocalized. So you're trying to figure out whether there could be the same causal variant underlying both those signals. And you're interested in that because you'd like to potentially make an inference that if this variant is causing variation in this gene, perhaps in fact it is this gene that is causing variation in this waist hip ratio adjusted BMI trait level. So that's the kind of rationale of doing colocus. It does not mean that this variant is working through this gene to affect this trait, right? So even though we say kind of causal, it truly is still this variant could be working through some completely different mechanism, right, to cause this trait. But this is for what we have, this is the best evidence. Okay. So the rationale, again, was to find not only genes, I'm going to show you only examples of genes, but it could be protein QTLs, metabolite QTLs, ataxi QTLs that may be causal for a disease. And then potentially to identify sets of traits that may have the same genetic underpinning. So everything I'm going to show you today is geared toward this gene molecular trait overlap with a GWAS trait, but we're going to work to extend this to GWAS traits. So our goal is to have a standalone browser that plays well with other browsers and to be able to display colocalization, as I said, from GWAS and the GWAS cis molecular QTL. Okay, so again, I'm going to have a gene-centric focus, but I think that we will extend this as time goes on into a more variant-centric, because gene-centric, I think, comes in part from pharma, from wanting to understand, what's the biology, but you could ask equally interesting questions about, which variants do you believe? So these are the questions. How much should this colocalization result, or a lack thereof, increase or decrease my belief in a gene of interest? And how much should I believe in the genetically predicted direction of effect between the level of a gene and disease risk? So these are some of the features that we either have in blue or are working on in kind of black. So ability to browse colocalized GWAS molecular trait signals and GWAS trait, assess the quality of these colocalizations, browse from multiple QTL studies. We just have to extend what we load, basically. Look at allelic series information using Mendelian randomization, find constellations of CMD traits associated with the GWAS signal from one of those, assess the certainty of the gene to disease link, and study the quality, kind of the quality assessment. Okay, so data required for colocus. So one of the things about this browser it's kind of a philosophical development, is that this assumes that fine mapping and colocalization data are available. And we've taken this approach in part because trying to do that is a whole nother huge enterprise. And also, I think it's good for the domain experts actually to look through and look at the data and make sure it makes sense. So the data that we would take in to be able to put data in the browser are colocalization results right now from Coloc, marginal QTL and GWAS results, fine mapping QTL and GWAS results. We support input from Apex. We're working on input from SUSE, and GCTA would be next. We have, and then some sort of study-specific LD, or it could be a reference panel LD. So these are the pieces of information that we need to be able to put this browser on. And what I'm going to show you, each row of the browser is a co-localization of one GWAS trait and one molecular trait in one tissue and cell type. And this current view is from an adipose, subcutaneous adipose meta-analysis, which is being performed by Corinne Small, Karen Mulkey, and I. All right, so when you come to the browser, this is the page that you would land on. So this page has a few different components. It has a filter part of the page where you can set parameters that you want to define the searches. And as I said, each row is a trait, gene, tissue, and it's also, in fact, a unique signal. So if there's more than one signal for those combinations, then there will be another row. So let me take you through the information here. Okay, so first off, you can specify the QTL gene. Let's say that I was interested in ANK1 or a genomic region of interest to be able to see the kind of parse down the information. The GWAS phenotype. So here you can see at this point, we've got 3 loaded in. You could select just one of those or a subset of those or all. And you can set the parameters that you'd like to see in terms of colocalization certainty. So this ranges between 0 and 1. A typical value that people would use is 0.5. You might want to be, so this is kind of the posterior probability that these things are colocalized with each other. You can also set an R² for the LD between the GWAS, the lead GWAS SNP, and the QTL SNP. So some people also like to set this. And then you can set thresholds for how strong you want the underlying independent signals that are being colocalized to be, so that you can either be more stringent or less stringent on what you see in the table. All right, so let's go to this table view. So in this table view, I've selected for waist-hip ratio adjusted for BMI, just one trait. You could have multiple traits. You can see I've put trait 2 here. In this case, when we're doing GWAS and gene, this is the gene name. So you can see VEGF-A here. All the tissues here are adipose, kind of redundant, but we'll have the capacity to load in, multiple different tissues. And then in the next thing, you can see that there are trait 1, which is your GWAS, and trait 2, in this case, your molecular, the gene. You can see that this is the lead SNP from the EQTL for that signal. And you'll notice there's a pretty array of colors, and we actually did this on purpose. So as you scroll down this table, if the colors are the same, okay, that means that the variant is the same. So what this allows me to do is to know that I'm scrolling down this table, because it's hard to parse SNP names, right? That all of these genes right here are co-localized to the same variant. And in fact, it's purple over here, that these 2 genes have exactly the same lead variant, whereas these 2 have actually the identical but slightly different variant. So you can see down here, there's a whole set of genes, right, that are co-localized to this GWAS signal. If you go over, next you see the strength of the negative log 10 p-values for the GWAS signal and for the molecular trait. I've sorted here, so you can sort each one of these columns and even sort on 2 of them if you want by a click down menu. I've sorted here on the GWAS values for the values for the waist tip ratio, adjusted for BMI. So you can see very strong. This is the p-value for the molecular trait. And then as I go over here, you can see the H4. So you can see strong evidence of colocalization here at 1. And then you can see the R squared. So if you look at the purple, this is the same SNP. So this is the R squared between the 2 traits. You can see a little lower value here. Most of these, what you'll see is most of these will be fairly high for strongly colocalized signals, but not always. So there are people who like to have both of these. And then this next piece, I'm not going to go into detail because we're going to simplify this, is simply trying to give you a sense of the direction of the effect signal in the GWAS and the direction of the effect signal in the EQTL when they're lined up with each other. But that'll be simplified. Okay, so that's our basic table. Okay, so now let's say I'm scrolling down through my table and I come to this set of results where we've got 5 different genes here that look like they're co-localized to this signal. So I click on one of these, okay? And what that does is it brings me to a next page where I can see the results at. So this blue bar here indicates the gene that I've clicked on. And this is our CBX3. This is the picture I showed you at the start, right? So you can see the underlying results. And then you can see on this side a locus compare plot, which just shows you that there is a beautiful concordance between these signals. All right. If I click on another one of these, then what you can see is this next gene, which is SNX10, looks equally beautiful. Okay, so it's beautifully colocalized. And in fact, if I clicked through onto other ones, you would see a similar picture. So this is allowing you to say visually, not just trusting that R squared of 1 and that H4 of 1, this looks like a beautiful signal. There are lots of genes with beautiful signals for this locus. Okay, so that's the main part of the browser so far. I'm going to show you more details of things later. So I want to step back for a minute and think about the data that will be available in colocus. So for the work that we're doing within the AMP CMD, there's going to be CA-QTLs, EQTLs from single nucleus data, and then there's going to be all sorts of QTLs from other existing studies in relevant CMD tissues. And this is just a list from different places of the data that we're hoping to integrate into this browser. So we really do expect that it's going to contain a large amount of information so that any gene that you put in, you might come up with data from multiple studies showing evidence of colocalization. Okay. So a little bit in the background of how this works if you're not in the AMP CMD, but you're thinking about this. So we have kind of 4 parts to this. So there's a genetic working group, a single nucleus working group, and these groups are working on producing data and then doing the fine mapping. So the molecular QTL, CMD, GWAS, getting the LD colocalization results. Then we have the knowledge portal. And the CMDGA, which is the molecular data, basically repository part of our work that will store and annotate and produce APIs so that someone can query this data we can query this data to be able to put it into the browser. We've got the development of the browser here and testing. And then we have the knowledge portal into which we're going to integrate the colocus browser so that when you go to look for information on a gene, you can if the information from colocus, some summary of it will come up, and then you'll be able to go to the browser to look at the actual underlying data. Okay, So making this work, and this is a little bit in people thinking about this kind of ideally. So you can think that when people do colocalization of a trait with a GWAS in a big consortium, you may have one canonical GWAS that everybody uses, um, and like one canonical set of signals, like identification through GCTA or some other method of independent signals. Ideally, in this browser, all of the molecular traits that are in here would be colocalized to the same version of signals from the same GWAS, because that would allow you to kind of do a head-to-head comparison across that. And so that for one GWAS locus, the same GWAS locus, you could show all the colocalized CMD traits, and you could show all the colocalized genes and molecular traits. So to do that takes some thinking ahead, and it takes some pretty broad agreement from people about what the canonical sets of things are going to be. If the data that goes in is colocalized to different GWASs that have been done at different times, right, they're going to have different signals. And so lining things up and really taking, I think, full advantage of this is going to be harder. Okay, so some planned additions to colocus. So the first one, I think you can see it and I can't up here. You see up at the top here. So we'd of course like to have information about the data that's in here. Right now it's a single study, not so important, but we would have tabs where you could go and look at the study characteristics of the studies that are in here, get references to them. We're working on building a QC module to look at the data actually that people put in, and then a gene summary page. So if you know that you're interested in ANK1, for instance, when you come here, you could simply go to get information on that gene. So I want to talk to you a little bit about the gene summary page. So some of the things that we've heard that people are interested in and have thought of is basically what would give you the most confidence in a gene and the most information about it. And this is in part from a pharma point of view, but also in part from a, just is this gene actually causal? So one is the presence of an allelic series. So that means for a given CMD trait GWAS and gene EQTL, is there evidence concordance in direction of the EQTL and the GWAS? And you have to have more than 2 signals, hopefully even more than that really you can test this with. Then thinking about gene specificity. So how many genes are colocalized to the same GWAS signal? So I've already shown you examples where there are multiple beautifully colocalized genes with the same signal. Once we get more tissues in here, thinking about tissue specificity of the gene, so the tissues and cell types that are colocalized with signals, it might be really interesting to see that in endothelial cells from muscle and adipose and liver, right, the same signal keeps coming up, whereas no other tissue seems to have that signal. And then also for a given GWAS signal, being able to see what other CMD traits are co-localized to it, so that you could get a sense of the constellation. Is this T2D locus where there appear to be other strongly colocalized insulin traits? Kind of what is the underlying biology that's going along with this signal? Okay, so if we think about the Mendelian randomization, so just to give you a sense and a little bit more information on how the browser works. So here you can see the picture you've seen before. There's actually 2 views that you can look at of your data. So one is the marginal signal that you see in your data. So this is just, what typically comes out of running your GWAS, running your EQTL. Okay. And the other one is conditional. So first I'm showing you the marginal signals for the waste-tip ratio and the RSPO3. You can see that they're pretty well in alignment with each other. Lots of different kind of LD. But if I go to the and click on the conditional picture, okay, so now what I'm seeing is there must be more than one signal, right, in this gene, because now that I have conditioned on another signal, you can see a very different picture here. This one still shows evidence of colocalization. And now you can also see another feature of the browser, which is that you see two SNPs over here, and we don't have them labeled yet, but this is the lead variant in theory from the trait 1, and this will be, if you click on it, the lead variant from the trait 2. Okay, so it's a purple diamond. You'll see it may not actually quite appear to be the lead variant, and this is a feature of something that is hard because we used for these analyses a predetermined SNP that was said to be independent. And sometimes when you use that to then do conditional analysis under other parameters, it doesn't quite come out to be the lead variant, or sometimes, not always, but that's why it doesn't appear to be the lead here. And then you can see the same variant is somewhere here. It's hidden underneath this SNP right here. You could also click on this, and that would allow you to see the lead variant in the other trait. So this is one signal. And then if I go forward, this is the other signal in this locus, so much sparser. So basically there's been conditional analysis to separate it out. So this locus has two different signals. They both look like they're pretty well colocalized. And so we would provide Mendelian randomization. So this is the picture of the Mendelian randomization, which is basically just looking at the effect size of the GWAS on one axis and the effect size of GWAS on one axis and the effect size on the QTL on the other, and asking if they're concordant with each other, so that you have an assessment, right, of whether you actually believe that the direction that you're seeing is relevant. I can tell you from some of the things that we've done, or that I've seen, that having just a single variant overlap with a single GWAS hit, and looking at when you have 2 or 3 or 4, not all of those directions, right, are consistent. So I think trying to infer from a single EQTL and a single GWAS signal for a gene that that's the direction of effect may be problematic, and that's why this is so valuable. Okay, so the next question is, how do we summarize that information, and what might you want to see? So I've made up this imaginary gene here as a potentially good target, the gene ABC. And here I've got 2 trait colocalizations, one to waste-tip ratio, one to type 2 diabetes. Okay, so the things that I'm going to look for here are number of signals colocalized. So here I like this. I got 3. I've only got 1 to T2D. So this is for a given gene, right? For this gene. Is there evidence of consistent genetic effect, right? Is there Mendelian randomization for this gene? Let's say yes, this one wasn't tested because there's only 1. Direction of association, same for both of these. And then this is the number of other genes colocalized. So in my imaginary case here, this is in fact the only gene that is colocalizing, unlike some of the other pictures where we saw that could have many. And then this would tell you how many other traits were colocalized. Okay, the next picture is perhaps of a potentially not so good target. I mean, it still may be interesting biological, but this is the gene CBA, made up. Again, trait colocalizations. This time, there's only 1 signal colocalized per gene, so I can't see if the effect is consistent, both positive. Number of other genes colocalized, 5 and 4. So this may be a gene that, if you're thinking of drug targets, you both do not have evidence of consistency, you really don't have certainty about direction, and you've got lots of other things that plausibly are colocalized here. You may still like this gene. It doesn't mean that it's wrong, but it just means that the genetic evidence is not so strong. Okay, so in summary, our goal is to display large amounts of molecular QTL, GWAS signals, colocalization, to facilitate searching, visualization, I would say assessment of colocalization results. You know, when you look at it, do you believe it? Because you can have things that come up with high probabilities of colocalization, and you look at them, you go, okay. And then to provide a context of colocalization results to help to contribute to the belief in the gene and its potential role in disease. Thanks.
Thank you, Laura. We have a few questions that have been accumulating, and then I'm sure people will want to have more questions. We have a lot of accolades too, like people like this. Let's see. So there's one question about whether this is public, and I know that it's not, but do you have a rough timetable of when it will be?
Oh, that's a great question. So what we're trying to figure out is how many bells and whistles we need to put into it before we make it public, because it certainly is under pretty strong, especially with the addition of Jeff Holtzman to our team, pretty strong developmental, I would say, undertakings. I don't know, Ryan, we haven't really talked about this when it would be, so I hesitate to say the answer to that.
Sure. We have another question about why only some of the GTEx tissues?
Why only some of the GTEx tissues? So we have not, so there's a really interesting question underneath there. So I know that there are efforts underway through QTLs to do all of the colocalization of the GTEx tissues. So this is really for tissues that we happen to have within the group, you know, of people who are within this AMP CMD. But the larger question really that you're asking is, wouldn't you want to have this for like all the GTEx tissues and everything that you could get a hold of, right, to really make this into a super browser? And I think that really that's only limited by being able to get the data and then handle kind of the results at that scale to still make it parsable. So I think we could think bigger. We've started out kind of small.
Yeah, that makes sense. Let's see, we have a question from Brian Palmasaro. Brian, you might want to unmute yourself and just ask it. It's about the conditional testing. Why does the conditional testing increase the p-values? Do you want to just ask that yourself? You might do it more clearly than me.
Yeah, sure. Thank you so much. I mean, this is very exciting. I've been just blowing up the chat with saying how excited I am to browse it myself. So thank you for all your efforts on this and sharing it. I was curious about the conditional testing because you showed kind of how the p-values increased and the locus zoom plot kind of was a lot more rich on the conditional testing. And is that just because you take only the significant hits for one trait? Like if it was for type 2 diabetes or waist hip ratio, you just take only the significant SNPs in that region and then only test those for EQTLs, so effectively reducing your multiple testing burden to kind of have a greater chance of getting a significant p-value? Or how does that work?
Do you have a specific slide that sparked your question? Can I,
just the one where you went and showed the effect of the conditional.
All right, let me bring that.
Locus.
I think it'll be easier to like.
Yeah, like what does the conditional, like how is that different than the marginal?
Sure. So let me go back to the marginal, because the thing that might be confusing you is that the scale changed between the pictures. So here's the initial, but there's a really interesting thing underneath your question. So here's the scale of p-values in the marginal p-values. So you're up to about negative log 10 of 200, negative log 10, I don't know, of 70 here. Okay. This is the conditional p-value. So essentially, when you're doing the conditional analysis, the first one that I'm going to show you is almost certainly conditioning on this variant. Okay, the next picture, right? This has the strongest signal right here. So and you see there's all this like herd of SNPs down here. Okay, so when you condition, I'm guessing you condition kind of, well, if you're doing it stepwise, it would be on this variant, right? So when you condition on that variant, watch the axes. All right, much less significant, okay? So this, I think, was that signal, that kind of herd of blue down there, all right? The next one that I show you is now going to be when you condition on the lead from this signal, right? It's kind of the reciprocal, right? So when I condition on this signal, now you can see something interesting. So if you look at this axis, that was 200 before, now it's 80. Okay, so what it says, and this is the trick, the hardest, the thing that makes interpretation hardest is that often lead signals, right? They're in the same region. They may be on the same haplotype. And when you condition on the haplotype, if they're sharing, like the direction of the signal is in the same direction, then it's going to go down because essentially it's being inflated, right, for both. You can have situations, and that's why I had to come look at this, because I hadn't looked at it, where the signals go in the opposite direction for the two SNPs. So when you condition on the one, the other one actually shoots up because you freed it from, right, the signal that's going in the opposite direction. And this doesn't happen to be a case, but it does happen, and that's why it happens. There are cases actually where there's a p-value of like 0.5, and you condition on something else, and it goes up to 10 to the minus 8th, 10 to the minus 20th. But it's because you've taken out an opposing, a SNP on the same haplotype in an opposing direction.
Thank you so much for explaining that. Yeah, that makes a lot of sense.
Yeah. Okay. Good question.
Let's see, we have a question from Andrew Patterson about the Mendelian randomization. Are you using the marginal effect sizes or the conditioned effect sizes?
Good question. We're using the conditioned effect sizes, and I think that those are the correct ones. They have all sorts of issues kind of hinted at the question from Brian, because it really depends on who you're conditioning on and what's tricky, and I've only realized this actually recently in thinking about this, is that when we do our conditioning in GWAS and in the QTLs, we're usually doing those completely blind of each other. Like the GWAS people go do their conditioning and we go do our conditioning, and then we come together. But if we haven't conditioned on the same variants, right, then the conditional signal also still probably has some residual, right, confounding in it. And we have examples that I can't show you here, where it looks like the GWAS signal has two signals hidden within it because it's just pretty small, right? But the EQTL, there's two different EQTLs co-localizing to the same darn GWAS signal. And it's because there's like subsets of that signal, you know, that brings up each. So I'd be curious to hear about what you think is best practice there, Andrew.
I actually don't know, which is why I asked. Just to be clear, the effect sizes you're using are from a single model where both SNPs are in, and you're using those jointly conditioned effect sizes. Is that right?
Yeah.
Yeah.
So
which we usually wouldn't get out of a GWAS result, because we're putting that other SNP in as a conventional covariate, and we're not getting the estimates for it.
Right. So you're in these, you know, in whatever program you're running, whether it's SUSE or APAX, which we've run for these, you get out the conditioned signal, right, from that. But the marginal signals, I think, just does not seem like the right thing to me, because there's so much LD confounding there.
Yeah. Thank you.
Yeah, I think there's also an issue within when you're doing Mendelian randomization of exactly how much LD you're going to allow between the signals that you put in as quote unquote independent. Right, and if you haven't separated, there's another issue there too.
Okay, and Kim Lorenz asks, sort of where will all the data come from? If I have a bunch of colocalizations, will you include them, or are you doing them all yourselves?
So we are pretty certain we do not want to do the colocalizations ourselves. We want others to either do them systematically or within that. So we will, all of the colocalizations for this AMP CMD will go to the CMDGA, which is the repository for the AMP CMD. So they'll be read in, we'll make sure that they're consistent in a format that we can take, and then we'll use, an API to take them down into our browser. So we want the 4 things that I listed on that first slide as being necessary for us to be able to display the results. Okay.
We have also a question from Jiang Li. How to deal with the direction association of a number of traits? I guess if the direction of association of a number of traits is inconsistent from MR locus of a single gene.
So let me go to the.
Okay.
Do you want to speak out your question? Because I'm not sure if I've understood it.
Yeah.
My question would be, like, one of your slides showed the direction association number of traits and genes are positive. The evidence of consistent genetic effects are not tested. Why you have a 2 independent locus, they all associate with the traits and the EQTL, but the direction of that association is inconsistent.
Yeah, okay. So that is a really interesting question, I think, for the Mendelian randomization, right? So just to be specific here. So let's say we have a GWAS locus that has 2 signals and an EQTL that has 2 signals. They're co-localized with each other, right? But the one direction says expression up, GWAS trait up, expression up, GWAS trait down. Okay, in the Mendelian randomization, that's going to get thrown out, right? Because you're looking for consistency.
Mendelian randomization, actually, most of the time they do the meta-analysis and regression. So I guess maybe to pick the meta effects, right?
Say that again, please.
So for the Mendelian randomization, like MR regression, if you have identified multiple independent locus, you do the meta-analysis. If the direction was opposite, but finally get the meta effects. For here, I'm not sure whether you have that consideration in the model.
In the MR locus, you do. I mean, I'm only superficially familiar with MR locus, but what I'm sure is that just like any other method, it's looking for consistency of direction. Okay, so the question is, if you see inconsistency of direction, makes that locus maybe harder to talk about. It is completely possible, right? Yes. That the relevant things that gene does back in embryogenesis, right? Yeah. There's a different direction of effect that affects, right, what's happening with that trait than what's happening, right now in an adult. So it's possible that there is 2 things going on. One is there's actually biology where at different times in different states, in different whatever, you are going to be right in a different direction. The other possibility is that you've got a lot of SNPs in LD, right? And in fact, it's a different SNP. This is not a causal relationship. You got 5 SNPs in LD01, R²1. It's a different, even though they look like they're colocalized, they're not, right? It's not causal. So it's just the wrong, it's a false, it's like a false flag there. So there could be something true, and that SNP should not even be in there, but you don't know it. So it's I think nice when Mendelian randomization is consistent. I'd say it's even nicer, like if there were 4 different SNPs or lots of SNPs, not just 2 that are consistent. Inconsistency is inconsistency.
So Brian has a comment that I think is relevant to this particular discussion. Brian, do you want to just speak it?
Yeah, sometimes we use the directionality variation. So you basically can plot the effect size and the direction of the EQTL. It's sort of like a locus zoom plot, but it's a similar idea, is that you just plot the directionality of the EQTL and the effect size along, against GWAS, whatever GWAS you're interested in, with the effect size and the direction. And you can sometimes see a linear plot. That would be like the best case scenario, and that tells you, it's sort of like this. Sometimes it it goes negative and positive.
A 2 SNP, I should get a more SNP, but this was just the prettiest underlying locus zoom plots that I had out of the ones we had. Ideally, what you would see, right, as Brian is saying, you would see a SNP here, and you'd see a SNP here, and you'd go, there's a lot of them, and they're all going in the same direction. This seems really unlikely to happen by chance, right? Because I think it's actually not so unlikely that this would happen by chance if you were just randomly, picking out. I mean, there's some black, but I think even with 2 SNPs, you could still get chance intervening here pretty easily. I think once you get up to 3 or 4, you're getting a lot more confident, but you got to have big enough sample sizes separated well enough, right, to be able to get enough SNPs, that have come up in both your GWAS and your gene to have that. So big sample sizes for EQTLs, I think, is really important for trying to do this work.
Okay, we have a question from Ravi Mandla. He says, we're testing for co-localization with the new T2D meta-analysis that we cannot run fine mapping for. Can those results still be included on this browser?
Why can't you run fine mapping?
So this is a multi-ancestry meta-analysis, and we do want to run fine mapping on it, but I think that's a whole separate project that will take a long time to put together still.
So that you've got multiple issues there, right? You've got the fact that most of the studies underlying, at least the ones that we have right now, are European ancestry studies. So you're going across like European ancestry to different LD patterns, right, underlying your multi-ancestry study. So I think the consensus is that there's a large amount of shared, probably causal variance across ancestries. But most of these methods that are doing the colocalization are looking for, some sort of similarity of those patterns. So I think colocalization is possible. I think it's hard to know what is going on once you've separated out signals from one thing and not from the other. What you're going to end up colocalizing are when you have an independent signal in your EQTL that is colocalized with basically the strongest signal in your multi-ancestry, because usually sometimes the first signals are really strong. So in the profile of p-values, that signal is going to dominate in the marginal, right? And so you may be able to get colocalization to if you have it to the dominant signal in your GWAS, you are not going to be able easily to get colocalization to those signals that are underneath it. I mean, I guess there's a TWAS approach we're really trying to basically integrate all those in at once. You know, but those tend to have, those tend to identify lots and lots of signals, and I have less confidence in those. So we'd have to talk about it. I'm curious to see what others think.
Okay, we don't have other questions in the Zoom in the chat. I want to point out that Eva Fain has had a lot of comments about complex disease in general, and you may all want to go in the chat and read those, but they're not directly relevant to this, so we're not going to go through all of them right now. Are there other questions or comments about the colocus?
Hi, this is Doug Keel. I have one question. I know this
Hi,
is
Doug. I
called.
think you're muted.
Oops.
No, I can.
Now
I
you're
can.
fine.
Can you hear me now?
It might be me who was muted for a minute.
I know this is all about colocus, but I noticed in one of the tables you had the fourth hypothesis from the colloq, where it's a colocalization. But is there a possibility of displaying the other hypotheses 1, 2, and 3 that might be of interest?
Oh,
I know
the
it's
H1, H2, and H3.
yes.
Yeah.
Just to keep an, I don't know, it might go against the whole reason you're doing this, but it might be additional easy information to display on a table.
So that is a question that we've been thinking about in terms of design, as to whether there would be a default set of parameters, and then there could be a possibility to add more. We're constrained to the size of probably a lot of us, that kind of 13-inch at home, right? Some of us bigger, but for those of us who have the smaller one. And so trying to decide what is most important might be different for different people.
Yeah,
so,
could be an option, like you said. Yeah.
These have been great questions. Thanks very much.
Thanks so much, Laura, and thanks everyone for all this great discussion. We're almost at time. I don't see any more questions. Noelle, did you have any closing words? Maybe she's not on anymore. Okay. Well, in that case, and if no one else has any more questions, we just want to thank you again. This has been great, Laura. And please, if you have more thoughts about colocus, write to our help desk and we'll get the suggestions to Laura, and that would be great.
Thank you so much.
Thanks.
Thanks.
Thanks, everybody. Bye-bye.
Bye.